Okay, so just to remind everyone here that we want to give Dr. He a chance to explain what he's done in terms of the science in particular, but also other aspects of what he's done. So please, can you allow him to speak without interruptions? As I said, I have the right to just cancel the session if there's too much noise and interruption. And I just wanted to state a couple of other things. So first of all, that we didn't know the story that was going to break over the last couple of days when he accepted the invitation to come and speak to us. So we didn't know this story beforehand. In fact, he had sent me the slides he was going to show in this session, and it did not include any of the work that he's now going to talk about. It was sort of preclinical data, but nothing involving human embryos that were implanted. And I should also state that we're in a venue. We have very generous hosts, Hong Kong University of Hong Kong, and our hosts have a strong tradition of allowing free speech, and so we are complying with that tradition of free speech. So anyway, I would like to, if you can hear me, ask Jiankui He to come to the stage and present his work. I don't know where he is, so hence the... Okay. All right. Okay. All right. Okay. Please not too long. Sure. Try to mute.
Okay, thank you. First, I must apologize that these results leaked unexpectedly, taken away from the community of the full data being presented immediately in a scientific venue. And through a peer review process engaged before this conference. So this study...
It's very disturbing for everyone here if you're taking photographs. Keep standing still, you've already taken photographs, you don't need to take any more.
Okay. Thank you.
You've been warned.
Okay, so this study has been submitted to a scientific journal for review. I also thank the Associated Press, who we engaged months before the child's birth, out of a commitment to the accurate reporting the study's outcome from many points of view. I also thank my university, although they are unaware of the study's conduct. So, Winnie Herbert, I should also thank you for your statements now and your wisdom that shared with me, as well as a community for discussing this data and providing this forum. I'll give an overview of our data focused on human and monkeys. Despite wonderful progress in HIV therapy and access to this therapy, new infections remain 3 times higher than the UN AIDS 2020 target. So HIV remains a top 10 cause of death in several countries, in particular developing countries. For uninfected children born by an HIV-positive mother, who make up a large percentage of births in area of Southern Africa. The risk of being infected by HIV in the first few months of life is many, many times higher than other babies. This is a serious unmet need, as an infection's severity is often made even worse by discrimination. So a copy of a natural protective antigen against HIV infection is carried by as much as about 10% of the population in several European countries. This antigen led to the first functional cure of HIV and decades of clinical trials, making this natural small deletion in CCR5 gene one of most studied variations, and the CCR5 gene are one of the most understood genes. We first explored the impact of CCR5 gene knockout in mice to investigate multi-generation effect. Editing was efficient as expected. We established a third generation CCR5 lung mice, which we confirmed by Western blot and flow cytometry. Tissue pathology was normal in heart, liver, lung, and stomach. So, two common behavior assessments showed no difference. We then assess whether this set should get RNA against the human CCR5 gene could be designed. So we assessed 7. I highlighted one that occurred at exactly the start of delta 32 mutation. It has an MIT specificity score above threshold, predictive of having no off-target activity. A few previous publications has assessed the same or similar get RNA in multi-cell type, including non-viable embryos. SG4 induced the most efficient editing activity in a seiner and 3PN human embryos. Since this target site is conserved in the monkey genome, we could use the monkey as an animal model to assess the SGRNA further. We found injecting the Cas9 closer to fertilization promoted the most efficient editing efficiency. Consistent with the Cas9 require 10 to find the target loss and its degradation over time. The rate of fertile eggs forming blood cells was not affected by exposure to Cas9, which we observed across experiments. So sequencing of the blood cells quadrant confirmed earlier Cas9 injection also reduced the mosaicism. To look more closely on the mosaicism, we also sequenced every individual cell in several embryos. Editing appeared to occur at the 1, 2, 3 cell stage. On the assumption that Cas9 degrades quickly and requires 10 to find the right target, we explored a strategy to reduce the mosaicism by delivering a second injection of Cas9 to an embryo at the 2-cell stage. The development of eggs to the blood cells was not impaired. We expanded sample size, which confirmed this previous observation. We still observed the variation across parents and the cycles. We then looked to see if this protocol could translate to human embryos. We found, as others have reported, the Cas9 protein was the most efficient delivery format. Lowering the dose compared to the monkey embryo also improved the efficacy. Upon the advice I received after presenting early results at the February 2017, the UC Berkeley Genome Editing Workshop, we edited non-viable embryos and established 2 embryonic stem cell names. So both are bioanic lockout, and the karyotypes are normal. Embryonic stem cell marker expression was normal by staining and the flow cytometry. This embryonic stem cell also found all 3 germ layers during the 14-day EB experiment, which is a marker of safety. Another serious safety concern is off-target. Embryo editing targets a single or few cell stage of life. Any off-target would pose very serious consequence and extend potential through the whole body. In adult gene therapy, off-target are expected, but a less healthy problem. We assessed off-target initially by single-cell whole genome sequencing of embryo prior to the implementation. We used the isothermal MDA amplification method to minimize the false positive rate and for unbiased coverage. The Miltopov lab used the same approach. We take a step further by sequencing the parent genome to detect off-target risk sites that exist only for each parent and a particular embryo, but not in the reference genome. We created a pool of off-target risk sites by first collecting any sites mentioned in previous publication. We added the genome sites for the unbiased assessment of a potential cleavage site. And in silico prediction, such as the MIT CRISPR design, both the original and the 2018 updated version. Finally, we import the parent genome, which allow facing to improve sensitivity and detect the novel risk sites unique to each embryo, which may emerge from inherited indel or SNPs. So all these loci found a personalized pool on the order of 10,000 spots per embryo. We use whole genome sequencing to assess for the spots and validate any findings by single sequencing. I will review genome sequencing data with the data on the null and null as focus and end. Of the potential cleavage sites identified by the genome-wide unbiased digenome assay, none was observed in the whole genome sequencing data. And no activity was observed at the risk sites identified by the 2018 version of MIT CRISPR design software and the original version. We explore off-target in the HESC-cyanine generated from edited embryo. Although we didn't have access to embryo with the parental donor's genome, we identified one potential off-target. This off-target is in the intergenic region, although we cannot confirm whether this is an inherited or is due to editing. So here you can see the editing efficiency across 19 viable embryos from volunteers. So we performed PGD, whole genome sequencing across the embryo, and didn't identify the off-target sites. So in one embryo, we identified a 6 kb deletion at the on-target sites. It did not affect any gene but CCR5. The CCR5 gene's distance from other genes protect against the risk of large deletion. We detect the large deletion by assessing for the chimeric reads and the visual confirmation. Now I will focus on the Lulu and Lala's genomic data. We sequenced the genome of both parents to confirm the target site conservation and to support off-target detection. The mother was HIV negative, the father positive, with undetectable viral load. So the ICSI and the sperm washing was used to prevent transmission. At day 5, so we have few cells were sampled from blood site for PGD. We follow on these results during the pregnancy by cell-free DNA after the mother declined on amniocentesis. So Lulu and Nala were born normal and healthy, with outcome score 8 and 9. After birth, we sequenced several different tissues. So in this Mark and Grace's first IVF cycle, pre-implantation genetic diagnosis found 2 blood sites were edited. One was a biogenic frame shift lockout, which showed shortened the CCR5 protein, similar to the natural protective variation. Another has an in-free deletion in one allele. The deletion is expected to destabilize local protein structure in the nearby HIV binding sites. The parents were informed of implementation of this in related to HIV infection and reminded them the option to leave the trial without implementation or to choose the wild-type embryos. The couple elected to implement this embryo to start a 2-embryo pregnancy. In addition to the single data, we also report to the volunteer whole genome sequencing data. The reads cover that more than 80% of the genome. The whole genome sequencing identified one off-target in the MEG-based intergenic region far from other genes, with no coding RNA and transcription factor binding sites. The volunteers were informed of the risk posed by the existence of one potential off-target, and they decided to implant. After the mother declined amniocentesis, serial cell-free DNA of blood test didn't observe the intergenic off-target from PGD. So another cell-free DNA test found no larval cancer gene mutation. After birth, the deep sequencing of the cord blood, which is primarily the baby's blood, confirmed the editing pattern observed during PGD and cell-free DNA. Single sequencing also confirmed this observation. After birth, both MiSeq deep sequencing and single sequencing did not detect intergenic off-target observed during the PGD. This suggests it was an artifact of a single-cell amplification or a mosaic off-target that happened to occur in the few 12-blast cells sampled for PGD. For whole genome sequencing, we did 100x cord blood and 30x on the placental. No off-target were observed genome-wide. Neither were large deletion. We will continue to assess the effect of editing in the twins, including testing a blood sample for susceptibility to HIV infection at the P3 biosafety lab. We also further investigate the off-target effects and the mosaicisms across multiple tissues, and plan to monitor the twins healthy for the next 18 years with the hope that they will consent as adults for continued monitoring and support. Thank you. Okay, so.
Okay, so what we're going to do is have a little panel discussion, just the three of us, to make sure that the details of the science are well understood before we open it up to some general questions. Okay. So Matt is up here to help me with this. So Matt, do you want to start or shall I start? Okay. You chose to work with CCR5 because you felt that this was a valid first gene to target with this approach. But do we really know enough about CCR5 and its function? Because, as you said, that there are naturally occurring mutations. There are many, probably millions of people who naturally have mutations in the CCR5 gene. They're mostly northern European, or that's thought to be the origin of it. It's very infrequent, at least the delta 32 mutation is very infrequent in China, for example. So that could be that could reflect either that it never spread here from Northern Europe, or that it's selected against in China. So it's known that having mutation in CCR5 protects against HIV infection. But does it predispose to other complications. So West Nile virus, we know there's some evidence to suggest that increased that. And then just a long question, I'm sorry, but well, answer that one first. What about other things? There's also a suggestion that maybe influenza, so patients with CCR5 mutations may be more susceptible to severe effects of influenza, which would be bad in this part of the world.
Okay, so we choose the CCR5. It's for multiple reasons. So first, it's HIV. It's a lethal disease in several developing countries, and also this HIV exposed but uninfected children not became a new global challenge. And their study in Zambia, also study in China, show that those HEU children may get affected from 6 months to 18 months in this 1 year period, with a possibility of 0.5 to 2.5%. So that is significant number compared to the kids in general. And so for this gene, we have studied for decades, and there's multiple clinical trials on that. And also for the Western layer and other potential side effect of this, so during the informed consent, it was written down to inform them will be Western layer virus, not infections. And also in our 18 years old, even longer monitoring program, there's the Western layer virus detection regularly.
And do you want to follow up on that, or shall I? I want to follow up a bit more on that. So, so there's this, the CCR5 obviously must have a function in the immune system where it normally operates, which is nothing to do with HIV. The immune system, we know, has effects throughout the body, and that includes the brain, right? It's known to affect aspects of functioning of the hypothalamus, the pituitary, the other aspects of brain function. You cited your own work on looking at some evidence to suggest that there was no effect on behavior or cognition, but there has been another paper published a couple of years ago suggesting that actually mice with mutations in CCR5 have enhanced cognitive abilities. So that poses an issue, because have you inadvertently caused an enhancement at the same time as dealing with this? So do you think we really know enough about CCR5 and its role in the immune system to choose that as a first gene?
Okay, so the first is I against using the genome editing for the enhancement. Second, so the study you mentioned, I saw the paper, and I believe we need more independent lab verification. And also for the CCR5, we start this. Another reason is we should start with some simple, well understood single gene as begin a conservative first model, so that maybe in the future you can move on to multiple gene, more complicated genotypes.
Okay, well, Matt.
So thank you for your clear presentation. I think there's going to be some questions about some of the numbers in the process. So for example, how many couples have you consented? How many eggs have you obtained from each mother? How many embryos have you attempted to modify? How many then had the correct modification? And then how many were attempted to be implanted? And then how many actually gave way to birth? So what's the pipeline that you have done as part of this project?
So there are in total 8 couples enrolled for this study, and 1 dropped out. And for the remaining 7 couples,
Excuse me, and they were all similar in father was HIV positive, mother was HIV negative?
Yes, the enrollment criteria requires the father to be HIV positive and the mother to be HIV negative, and plus some other age requirement and others. So So for all the couples, they, after informed consent, for multiple rounds, informed consent with the scientists and also with the team members, and then under the normal IVF procedure. So collect eggs, and then we inject casein protein and get RNA.
And sorry, how many eggs total between the 7 couples?
So in total, we have about 31 polar cells, but that number is it's 30 embryo blood cells that yeah.
So 31 were injected.
It's more. I mean, what I mean 30, it's developed to blood cell stage, and with that, about 70% of the embryo were edited.
70% had biallelic editing, or 70% had monoallelic, and what was the percent mosaicism in those 30 embryos when you, well, that's right, you wouldn't know because you only took 1 cell.
Yeah.
So, and then, okay, so why did you decide on these 2 rather than the other 24?
This couple happened to be first to be pregnant.
Have you subsequently implanted the remaining in the 6 other couples?
So the clinical trial was paused due to the current situation.
I have one other thought. I'm from the United States, so I'm not completely familiar with how the review process. So how did you go through, who did you discuss this trial with in terms of your supervisors, mentors, other people, in terms of getting feedback on the trial design, the consent process? Tell me sort of the scope of the team that was involved in designing this clinical trial.
Okay. So when I start this, even from the preclinical study, I first talked to a couple of scientists and doctor to find out CCR5 is the one to recommend. And then once I have some early data on the preclinical, I presented in the Cold Spring Harbor Lab meeting in New York in 2017, and also in the UC Berkeley Genome Editing Conference. So some of the audience also in that conference too. So I get feedbacks, positive feedbacks, and also criticisms, and also some constructive advices. And I continued to talk to not just scientists, but also the top ethicists in the United States at Stanford. Like Winnie Herbert, as mentioned, multiple times talk. And also I show my preclinical data to many scientists visiting. When I started the clinical trial for the informed consent, it's we take the NIH guideline as a reference and drafted the informed consent, and later was reviewed by a U.S. professor. And when it's pregnant, this informed consent was reviewed again. So at a subsequent supplementary material for the informed consent to add the long-term follow-on plan. And also.
Let me go to that. How many people read the informed consent before you showed it to the family? How many people reviewed the informed consent and felt like it was appropriate?
So outside of my team, there are about 4 people. And when this couple was informed consent, there's observer from United States professor and also a Chinese professor in the Chinese Academy of Science, and it's audio recorded.
Yeah. So on the informed consent issue, was that gained by an independent person talking to the patients, or were you or your team involved in that process directly?
That's including the first round is team member went to talk to the volunteer first for 2 hours. And then after about 1 month, the volunteers came to Shenzhen. And I personally, together with another professor, give up 1 hour and 10 minutes informed consent.
But you were directly involved?
Directly involved. Because after 1 month, they actually bring out the papers, see off-target, CRISPR things, already information there.
And one more question, maybe even upstream. How did you recruit these couples into your study? Was it done by personal connections? Did your institute put out a release? So how was the recruitment done of these particular couples?
It's by a HIV and AIDS volunteer group.
Okay. I think what we should do now is start to open it up for questions from the floor, but David Baltimore wants to say a quick word first, if possible. Yeah. So then, when I come to taking questions, I will take questions from the general participants who are lined up. I will also I have questions from the media, so I have a lot of questions. Many of them are the same, so I'm not necessarily going to say who asked the question because they're the same questions. But actually also quite a few of them have actually been answered during Dr. He's talk, so I probably won't bother with those either, but I'll be quite selective. But first of all, David.
I want to thank Dr. He for coming and for being responsive to the questions that have been asked. I still think that the statement that we made at the end of the last meeting, which is that it would be irresponsible to proceed with any clinical use of germline editing unless and until the safety issues have been dealt with in this broad societal consensus, basically an open process, that has not happened and that it would still be considered irresponsible. I don't think it has been a transparent process. We've only found out about it after it's happened and after the children are born. I personally don't think that it was medically necessary. The choice of the diseases that we heard discussions about earlier today are much more pressing than providing to one person some protection against HIV infection. I think there has been a failure of self-regulation by the scientific community because of a lack of transparency. I'm speaking here entirely for myself. The committee that organized this meeting will be meeting and issuing a statement, but that will not be until tomorrow. Tomorrow. Why don't we continue?
Okay. All right. So let me start off with maybe David Liu from.
Hi, David Liu from the Broad Institute, Harvard, and HHMI. First, I'd like to echo David Baltimore's comments, thanking you for coming here under some unusual circumstances. I'll limit myself to 2 questions. First, I just don't see the unmet medical need for these girls because the father is HIV positive, the mother is HIV negative, you already do sperm washing, and thus you already could generate uninfected embryos that could give rise to uninfected babies. So could you first describe what is the unmet medical need, not of HIV in general, which I think we all appreciate, but what is the unmet medical need for these patients in particular? And second, you justify the critical decision of implanting these embryos to generate a human pregnancy with the decision made by the patients as opposed to made by the scientists and the doctors and the ethicists, can you also comment on what is our responsibility as scientists and doctors and independent communities to make that decision for the patients rather than allowing patients to make critical decisions like that seemingly on their own? Thank you very much.
Okay, so the first thing, guys, whether CCL5 is unmet medical need. So, again, I truly believe that this is not only just for this case, but for millions of this HEU children. They need this protection since the HIV vaccine is not available. And I have personally experienced with some people in some AIDS village where 30% of village people were infected. They even have to give their children to their relatives or uncles to raise just to prevent potential transmission. And also for this specific case, I feel proud actually. I feel proudest because the Mark thought he lost the hope for the life. But when the baby was born and with this protection, he sent a message at the day of birth, say, I will work hard, earn money, and take care of his 2 daughters and his wife for his second half life. So
can I ask, before we get to David's second question, you said that there's been no other implantations, but just to be clear, are there any current pregnancies with embryos that have been genome edited as part of your clinical trials?
there is another one, but it tend to monitor.
It's what?
There's another potential pregnancy.
I think you said a very early stage, so it's a chemical pregnancy rather than.
Yes.
But in the interest of the transparency.
Yeah.
Thank you very much.
Well, let's do this side first, and then I'm going to ask a question from the media, so go ahead.
My name's Evan Kirksey from Deakin University in Australia, and I have a two-part ethical question. So first, I was wondering if you could just slow down a little bit and talk about the institutional ethics process that you say that you went through. So looking to the past. The second part of my question is really about the future and how you understand your responsibility to these children. So your last slide indicated that you're going to be doing some follow-up treatment. So just an invitation to slow down a little bit and talk about your responsibility towards the future as well.
Okay, so just ask not you, but people here a question. So do you see your friends, your relatives who may have suffered from genetic disease? So what I said, those people, they need help. We should, for millions of families with this disease, inherited disease, or be exposed to infectious disease, we should show compassion to them. And if we have this technology, we can make it available earlier. That would be help much more people for this in need. So when talking about the future, what I mean is, first, that it's a transparent, open, and share what the knowledge I accumulated and to the society, to the world, and then let the society to decide what we should to do in that step.
If I might, my question was actually much more specific about the actual children, not an abstract question about the future, but going forward with these children that have been born, how do you understand your responsibility to them?
I mean, it relates to actually questions from the media, a set of them, which is basically, questions like, will you publish the identity of Lulu and Nana in the future? How are you going to prove the effectiveness of the treatment if the two individuals remain secret? But of course you have this conflict between you really have to protect patient identity in this case, but the world will want to know whether they are healthy, whether the method has any negative consequences or positive consequences. So how are you going to deal with that?
So first, it's against the Chinese law to disclose the identity of the HIV positive people in public. Second, so for this specific couple, it's under careful monitoring of health, and I think I would propose that it's the data or the information should be open to necessary regulatory and or necessary to maybe a civil panel of experts.
Mm-hmm. Okay. Okay, let's get a question over the side. Yeah.
Thank you very much. My name is Jiankui He. Beside my work, I also as a founding member of International Law and Technology Interoperability Association. We discover about law and technology and the future of legal profession. There are a lot of questions has been collected from our members, but I only ask a few globally.
One question, please. Choose a good one.
Okay, the good one, I think everyone is great. So the most important one is like, we are curious, how did you convince the parents when you started this experiment? Did you tell them like there's alternative solutions, for example, other ways to avoid of AIDS infection of their child? And second, it's about how did you done the ethics review? How many institutions has been involved, and how was the process? Thank you very much.
Okay, so the first question, how we convinced the patient. These volunteers, they all have good education background. So they know pretty much a lot of information about the HIV, the drug, all this alternative approach to them, or even the latest research articles published. This is common, as all the HIV-infected people, they are usually in a social network together, where the latest advance in HIV prevention treatment information is available. So when the volunteer come to the informed consent, they already understand quite well about the genome editing technology and its side effect or potential benefit. So I think it's a mutual exchange of information that made the volunteer made the decision.
Can I ask a question again that goes back to transparency? Would you be willing to post the informed consent, obviously the generic informed consent, and your manuscript in preparation in a public forum so it can be reviewed, such as on bioRxiv or on a publicly available website for the informed consent so the community can read in detail about what you've done? Would you consider doing that?
Yes, actually, the informed consent is already on the LAMP website. You can just search my name, you will find it. I have an English version there, so you can read it. Mm-hmm. Second, for the manuscript, when I draft the manuscript, there are already about 10 people outside my lab. A few in the United States help me to edit the manuscript. When it's ready to submit, I also send out for several to give comments. So I originally want to submit to bioRxiv. It's exactly my plan, but some advices from some people that I should go peer review first before it's posted on bioRxiv. Yeah. So I took that advice.
I think you took that advice, but would you change your mind now? Because I think the circumstances have changed, and I think, as you can see, there's a big demand to know exactly what you did. You don't have to answer that now, but just to say, you should think about that. Let's take a question here first, and then I'll do another one from the media.
Hi, I'm Anna Middleton, and I'm Head of Society and Ethics Research at the Wellcome Genome Campus in Cambridge. I'm also a genetic counsellor, and I'm very interested in the informed consent process. So if I'm understanding you right, there's a consent form that you're happy to share now. It was reviewed by 4 people, and there was a conversation that lasted about 10 minutes with the patients. Given that we know, particularly in the UK, that the average reading age of the general public is around age 10, and that the vast majority of the public don't understand what the word genome is, I'm quite interested in what happened in that conversation and how you explained what the risks were and what evidence you have that they actually understood.
Okay, so yeah, I can describe that. First, I don't know if Kia correctly, you're saying 10 minutes, and it's correct, it's 1 hour and 10 minutes
1
for
hour
this
and 10
couple.
minutes, okay.
So what happens, it's in a conference room with these couples, me and 2 observers. And the printed copies was given to the couple before the informed consent.
Do you know that they could read them? Did you know that they could read them and understand them?
I didn't get that.
Did you know that they could read okay and understand what you were saying?
Yeah, they are very educated. Yeah.
Okay.
And yes, so and then, so each of take this informed consent. I explain from page 1 to page 20, line by line, paragraph by paragraph, and they have the right to ask any questions during this informed consent. They get, and so they, yeah, once we go through the entire informed consent, at the end, I leave them to private discussion. So you have freedom and time to discuss the couples, and also they have the choice to decide today, or you could take it home and decide later.
So were any of the team actually trained in taking consent, or was this the first time you'd actually had conversations about this?
I'll start again.
Were any of the team that took the consent, were they trained in the process of dealing with consent issues? Were they trained at doing what they were asked to do?
So I said I have 2 rounds of informed consent. The first round is a team member is supposed to it's informal. It's just informal conversation for 2 hours. Talk to this person to explain. The second one is a formal one. It's I personally explain that. So I do read the guidelines from the NIH on the informed consent, even when we drafted the informed consent.
Okay, thank you.
Okay. Question from the media. Can you please explain the source of the funding for this work?
Okay, so when I started this, I was a professor at the university. So 3 years ago, when I began this, the preclinical study, the university paid the salary and all the regions, so this for preclinical study. So when move on to clinical study, all this patient's medical care expense was paid by myself. And a small amount of sequencing cost is covered by my startup funding in the university.
Okay. So there was no funding from industry or from a company, because you have, just to make it clear, you have an involvement in a company, but that was not involved in this project. Is that true?
Neither of my company was involved in this project by providing funding, people, space, or equipment.
And what about, did the families pay anything, or were they paid anything? So was there any exchange of money?
So it's all written down in the informed consent that we pay all the medical expense, and they are not receiving much extra money for that.
Okay. So let's have this side, sorry, questions.
Hello, Dr. He. I'm Li Mengyin from the University of Hong Kong. And since there is lack of the evidence to delete this kind of gene-making silence in human, and as a scientist who should be responsible for the patient, may we know your future medical care plan for these babies? For example, how would you give them the vaccine scheme? And also, the more important thing is, how could you evaluate their potential mental health? Because they have to be under the very strict or abnormal supervision in your team. Thank you.
How are you planning to monitor the medical health of the babies in terms of vaccinations and neurodevelopmental outcomes?
Yeah, so with the long-term the healthy follow-on program, the informed consent is posted on the website of my lab. So it's clearly the state on which each year what examination will be performed, including development, neurals, regular blood tests, all these informations, and plus the HIV infection, Westdale infection, all this information. So it's all available in the informed consent.
Okay,
Sorry,
thank you.
okay. Question here then.
All right, Wen Shenwei from Peking University, China. I have a technical question regarding the off-target assessment. You mentioned you did this single-cell whole genome sequencing. As far as I know, there's no reliable or mature technology to conduct the so-called single-cell whole genome sequencing. So this is the technical question. Second is, there's a consensus to not allowed to conduct genome editing on germline cells. This is consensus between the international community, including Chinese community. I assume you are very well aware of this. This is a red line. Why you choose to cross this line? Hypothetically, if you don't know, why you perform all those clinical study in secret? So can you explain?
Okay, so first about the off-target by sequencing. So before implantation, we can only biopsy 3 to 5 cells from the processed. From that, we amplify the, for the single cell, it compared to book cell sequencing, we'll get a less coverage on the whole genome. So for the book cell sequencing, we may get 95% of genome coverage. For single cell, the number of cells get 80 to 83% of coverage of genome. So that's the current state of art. And that means that there may be off-target missing. But we gain the confidence. It's not just look at this embryo. That means we already did many of them. By combining all this data together, we get to understand how much off-target will happen. So
the second part of the question, which is also questions I had from the media, which is essentially why so much secrecy around this, particularly when you know that the general feeling amongst the scientific community is that we shouldn't go ahead yet in doing this. So why is there so much secrecy from, for example, the Chinese authorities? So you know the accusation now is that you've broken the law. So if you'd involved the Chinese authorities as you were doing this, they may have said you can't do it, but you went ahead without really discussing with people. That's how it seems.
Okay, so as I said, I have been engaged in the scientific community 3 years ago, publicly speaking in the whole Spring Harbor about previous data, and in Berkeley, and also in Suzhou also has a whole Spring Harbor Asian conference, and constantly get feedback from them. When I moved on to clinical trials, I also consulted with several experts in the United States about ethics and also this sciences. So, and also the Chinese people there.
Okay, well, is it Maria next? Sorry, yeah. Maria next.
So, Maria Jason, Sloan Kettering in New York. Immediately, for me, it raised many scientific questions, but also a very personal dynamic for these two girls. So that I think if things had been thoroughly vetted, there may have been a proper discussion of. But for example, there are two sisters, one of whom I assume now is refractory to HIV infection, and that was a desired outcome from the parents, or at least the father you specifically highlighted. So within the family dynamic, are these two daughters going to be treated very differently? And then the other side of that, or related to that, is the one girl that is refractory to HIV infection, is she now going to be desirable for breeding, for, you know, will this change her whole dynamic in terms of marriage and children, because a spouse may be particularly interested in getting this mutation within the family? So, you know, even this very simple thing with these two girls being different in the family, and now this being something of an enhancement, preventative trait, not just a disease correction, but now something new that could be introduced into the population.
Yes, it's more on ethics or philosophy thinking of this. So I have reflected deeply on this. That's why I published a 5 core value for genome editing. That's including, specifically related to what you're talking. First is respect the children's autonomy. So we're not using any these tools to control their future, their occupation, or what they're going to do. And it's to give them freedom of choice. And the second one is, we say it's a gene don't divide you. It's the children we sort of encourage them to explore the full potential and pursue their own life. The life is right by the children themselves.
But for 18 years, they're children, and they don't have that autonomy. Now their genotype may quite affect their upbringing.
Mm-hmm. So do you think the fact that they've been genome edited is going to affect how they see themselves as people?
Not even perceive themselves, how their parents perceive them, how their relatives perceive them.
They will know, presumably at some point, that they have been edited, so it's going to be very unique.
I don't know how to answer this question from my eyes.
No, okay. I think, so I'm being told that we're going to have to stop the session very soon. I'm just going to ask, I'm really sorry, but I think we'll have to, I'm just going to finish off with 2 questions from the media, which are, again, some of these have been repeated several times, which is really, well, first of all, did you expect all this fuss? Did you expect all this reaction? Because you were, you know, even if you had managed to succeed in your aim of having the paper, you know, reviewed and published first, there would have been a lot of fuss at that time as well. So did you anticipate this?
It's because the news leaked out, it became all anticipated to me. Because my original plan or original thinking is based on the survey of the United States or the US, the British, also as the ethical statement that, or the Chinese survey that give us the signal that the majority of the public is supporting using the human genome editing for treating, including the HIV prevention.
And then the very final question. So, okay, if this was going to be your baby, would you have gone ahead with this?
Okay, that's good. So if it's my baby may have the same situation, I would try it first.
Okay. All right. So I think we should thank Dr. He for his appearing here, and thank you.